In this screencast, we're going to talk about the genetic code and what that means and why it's so important. So first of all, the genetic code is basically that the amino acid sequence of polypeptides, it's going to be determined by the mRNA according to the genetic code. I mean, ultimately it was the DNA, but you see how we have every three nucleotides of mRNA will ultimately is what we kind of rely on to be able to determine which amino acid will be added to the growing polypeptide chain when we're doing this whole translation bit, right? So every three, you've got the codon, the anticodon will bring down the amino acid when they attach. So this sequence of amino acids is determined by the mRNA, the genetic code. All right, so and one thing that's interesting is each codon is going to code for only one specific amino acid. So UGG will always code for tryptophan. Okay, so this UGG will always code for that. Now, granted, we know that there's tRNA in between, but this is, if we were to read the mRNA strand, we know that this would always code for tryptophan, just like AUG would always code for methionine. So every three, it's only going to code for one specific amino acid. However, as you'll see when we're looking at the mRNA table, that sometimes there's more than one codon that will actually code for the same amino acid. So just because UGG codes for tryptophan doesn't mean there's not another codon that also codes for tryptophan. However, UGG will always only code for tryptophan, not another amino acid. Okay, let's take a look. So here's an mRNA table. And so when you have a sequence of mRNA, you can go through codon by codon, and then you can translate that into the appropriate amino acid using this. So the way you use the table is you have the first mRNA base is this. So you start on the left-hand side, and then you look at the second base, comes down in the columns. And then once you have the right row and column, you can look within for the smaller rows inside the larger rows to find that third one. So 1, 2, 3. Okay, so let's look at UGG. We already know that it's tryptophan, but let's take a look and make sure that that looks right on the table. So U is this first one here. So we know it's going to be in this larger row for U. And the next letter is G. So I'm going to go all the way to the column of G. So we know it's going to be one of these four. And then the final one is the G. So it goes right here. Okay. So UGG gives us the tryptophan. Now, I want you to hit pause and see if you can translate the rest of this, the UUU and the GGC and the UCA. Hit pause. And when you're done, come back. And then you can see here's what it ends up being. So UUU would be U, so it's here. And then the final U would be here, so phenylalanine, right? GGC, so we have G in this larger column, G, and then the C, glycine, and then the last one, UCA. So here we go, U, C, and then A. So we get serine, okay? All right, so you need to be able to practice with that and always pay attention. Now, sometimes it gets tricky because sometimes what they're going to do is they will give you the DNA template strand, and you have to determine the right amino acid. So then you have to transcribe it and then translate it. Sometimes they might give you the tRNA, so you have to backtrack to the mRNA to use the table. So always pay attention to what the strand is that they give you. So if they give you the mRNA, you can go straight to the table. If they give you the DNA, maybe they'll give you the sense or the antisense strand. So pay attention, get it to make sure that you know what the mRNA should be, and then finally you can use the mRNA to translate it. Okay, so next thing I wanted to actually bring up is the fact that this genetic code, which codons code for which amino acids, that is actually universal. And so that means that we can take a gene from one organism, like a jellyfish, put it into a pig, and the pig can express that particular gene as long as the environment has all of the things that it needs. So, and putting jellyfish genes into organisms is actually kind of a common thing in biotechnology. We'll even give it a try, trying to put a jellyfish gene into bacteria, and then the bacteria can express that gene as well. So you can take the genes from humans, which is super handy, like the insulin gene, and we can put that into yeast or bacteria, and then the yeast or bacteria can build the polypeptide chain to ultimately make the insulin. And so that's really important in biotechnology because that's actually how we get a lot of proteins that we need to manufacture for health, such as people who need to inject insulin. It's much easier to be able to produce that through biotechnology than it is to try to extract it from other organisms. So now we actually put the insulin genes, we splice those into yeast cells, and then they can help create that insulin that we then use to mass manufacture for people who have diabetes. All right, one last thing I want to share with you is that a tRNA molecule has a couple of different sites. It's got, this is where it's going to have the amino acid attached, and then here's the anticodon. This is kind of a picture you might want to recognize some of these cartoons of tRNA. And then this anticodon will always correspond with the attachment site because there is an enzyme called tRNA synthetase, you could just call it tRNA synthetase, that actually is specific to the specific tRNA molecule and the specific amino acid. So when the tRNA and the amino acid join into this particular enzyme, these are the two substrates, then with some ATP, it will join them together. And so that's how we know that the anticodon will only have the specific, or this tRNA will only have the specific amino acid attached to it because it has to fit the active site of the particular tRNA synthetase that will put those together.